Hi everyone, in biology you'll know that single-celled organisms like an amoeba can use diffusion in order to obtain the oxygen and the nutrients they require, and that's because they have a large surface area to volume ratio. Humans are multicellular, they have a much smaller surface area to volume ratio. Diffusion is therefore far too slow, and therefore they require a circulatory system consisting of a heart, which acts as a pump, blood, as well as vessels, the arteries, veins and capillaries. Today's experiment is all about using agar jelly to look at rates of diffusion, and by comparing small agar blocks, they're representing the amoeba, with large agar blocks, they represent the humans, we'll really be able to unpick the science behind this. So in order to study this, we're going to use agar jelly, which has been stained using cresol red. It has turned pinky red colour, and it's done that in the presence of alkali. We're going to use our ruler and our knife to cut small blocks of agar jelly. So here's my very small agar block jelly. This is representing our amoeba, and it has a very large surface area to volume ratio. This larger block over here is representing our human with a very small surface area to volume ratio. So I'm now going to fill up these glass bottles with some very weak hydrochloric acid. My next stage is to place each agar block in turn into the hydrochloric acid. I'll use the stop clock to time how long it takes for the red colour, the pink colour you see here, to turn yellow. As we can see, look, this is almost decolorized, showing how very quick the hydrochloric acid is soaking into the jelly. This one's taking much longer because it has a much smaller surface area to volume ratio. Look, it's basically decolorized. We compare it with this one, much slower process. So this really proves why diffusion is far too slow in large organisms which have much smaller surface area to volume ratios. Let's just do some maths to back up our findings. So we're going to start with the smallest cube over here. So remember, to find the surface area, you do length times width times 6. And that's because if I draw a cube here, if you count the number of faces, it's 1, 2, 3, 4, 5, 6. So that's why you multiply by 6. So in effect, that's 2 by 2, which is 4, times by 6, which is 24. To find the volume, it's more straightforward. You do width times length times height. So that's 2 times 2 times 2. And then the surface area to volume ratio, you just do surface area divided by volume. Let's do the next one now. 4 times 4 is 16. Times 6 is 96. The volume is 4 times 4 times 4. And then we'll do a third one. Surface area of the next cube is 6 times 6, and it happens to be times 6. The volume is the same. In terms of looking at the time taken to decolorize, the smallest cube took the shortest amount of time. It took 18 seconds. Followed by this cube, which was 34, followed by this one, which was 52 seconds.